So today we're very fortunate to have, I think, one of the bright young people in the field of bioinorganic chemistry. I'm a little bit biased because Melissa received her PhD at the University of Michigan working on de novo protein design and had as her first paper one in Nature Chemistry talking about the best de novo designed enzyme that had been made to date. And quite frankly, it's still up there among the best, if maybe not the best. Um, she then went from my research group to Steve Lippard's lab at MIT, where she did a lot of work on zinc sensors. And after finishing her postdoctoral stint there, she joined the faculty at the University of Houston, where she has had her independent career for about 3 or 4 years now, I guess. Probably seems like forever, and yet not very much time, especially given everything going on with COVID. So Melissa's interests are now in metallobiochemistry of the gut. And what we're going to be hearing from her today is an overview of basically how everything we've been talking about, from siderophores to copper and zinc and calprotectins and whatever else, play in the gut biota, how the metallome is important to that. And this will also serve as a preface to the lecture that Professor Carver will give later in the term when she's talking about uh, the metal levels and infectious diseases. So with that, I will pass the baton over to Melissa, and I'm really looking forward to hear what you have to say.
Okay, thank you, Vince. Thank you for inviting me to join in this fun series for the course. So hopefully my screen is showing up properly now.
Yes, it is.
All right, and so today I'm gonna give really what is sort of an overview of this emerging area of bioinorganic chemistry, which is understanding how metals behave in the gut microbiota, so metal ion trafficking and metal homeostasis. And so to give you an idea of what I'm going to talk about today, I have a little overview. So we're going to go through some of the important aspects of the microbiota's function and its structure, as that will relate to how metals are moving about in the microbiota and how they're controlled and how they're affecting the microbiota. In this field, a lot of the studies have focused on iron and zinc, more so iron than anything. And so a lot of what I'm going to talk about is going to focus on iron and zinc. There have been some limited studies on copper and manganese, as well as some studies on non-transition metal ions like calcium and magnesium. But I thought I could give a better story focusing on iron and zinc and give you a good overview just focusing on these two metals. So I'll do that and cover just some general background that we need, which is how these metals are absorbed in the gastrointestinal tract from our diets, as well as how the bacteria take them up. And so some of these are going to overlap with some of the things that you've already seen and will see, but it'll help put it into context. And then with all that in mind, we're going to discuss what are the different factors that can influence metal availability in the gut. And then two things, how metals affect the microbiota, but also how the microbiota can affect the metals. So both metal availability and homeostasis. And then hopefully we'll have time to cover a little bit in terms of what tools we can use to study metals in the microbiota. And so this is directly related to my area of research. Okay, and so the human gut microbiota is a complex and dynamic collection of microorganisms. It's also the most densely populated community in the human body, community of microbes. It's primarily bacteria. They make up only about 0.3% of our body weight. 65% is our own cells, since they're much smaller, but they have nearly 500 times as many genes as those that are found just in the human genome. And so given that contribution, they play a lot of important roles in our health. So they're crucial to our health. They do things such as facilitate our digestion. They synthesize vitamins that we need. They're important for immune system development, as well as protecting against pathogens. So by either secreting antimicrobial compounds or just by being there, preventing pathogens from coming in. Okay. And so a little bit more about the makeup of the microbiota. It has temporal and spatial variations in the composition. And so can you confirm if you can see my arrow?
Sorry, go ahead and do it one more time.
It's here. I might have to check
It's
something.
very light.
Yeah, you have to go to the bottom corner. There you go.
I got it. Okay.
Yep.
And so the density of microbes actually increases as you go down the gastrointestinal tract. So it's quite low in the stomach, increases through the small intestines, which are kind of this light pink here. And then there's high densities of bacteria in the large intestine. So this is just in terms of CFU, which is colony forming units per gram of intestinal contents. The composition, so the species that are present, there's hundreds of them there, is affected by things such as the nutrient availability and also environmental factors. So age, health status, inflammation, antibiotic treatment. So there's a lot of complexity here. There's also a variation when you go from the lumen, so sort of the center of the gastrointestinal tract, to the mucus layer and our cells, so the epithelial cells. So the mucus layer protects the epithelial cells from the bacteria that are in the central area. And so the bacterial density decreases going in that direction. So some bacteria can actually embed themselves into the mucus layer. They have proteins that allow them to adhere to it. And some can even use it as a nutrient source. And so the mucus layer it protects our cells, and it's primarily composed of a protein called mucin, which is a glycoprotein. This is secreted by goblet cells, which are interspersed here with the epithelial cells. And it's basically a polymerized glycoprotein. So I've got a little cartoon here showing the sugar groups on the protein. And so some bacteria have proteins on their surfaces that can actually facilitate their adherence to the mucus layer. And so that may give them an advantage for colonizing in the gastrointestinal tract. So if they can adhere better, they might stick around longer. And in the case of probiotics, this may help them be there to do their beneficial functions a little bit longer. Bacteria can also adhere to some other sites, such as extracellular matrix components on the cells, like collagen. And you can also think about the bacteria adhering to each other as another aspect of colonization. So if they can aggregate and potentially even form biofilms, although mature biofilm formation in the healthy gut has not been detected. All right, and so what about the metals? So of course, in this class, you know that metals are important, crucial for all organisms. This, of course, includes the microbiota. And so the members of the microbiota have to get their metals from us or our diets. They manage to do this without harming us, at least under normal conditions. And so just like metals need to be tightly regulated on the cellular level, they also need to be well regulated on a multi-organism level in the gut microbiota. And so from that perspective, we're really interested in identifying what these mechanisms are. And in order to do that, we need to have a good idea of what metals are there, in what amounts. And how they're moving between species and between the host and the microbes. Okay, and so you've seen a little bit on metal homeostasis already. And so this is just a eukaryotic cell with another look at zinc regulation, where you have both uptake and efflux systems, as well, so ZIP and ZNTs transporters, as well as transporters that can shuttle the zinc in and out of different intracellular organelles. And then of course, they can also bind to proteins. And so this is a general example in eukaryotic cells. And what we're going to talk about over the next few slides are specific uptake mechanisms for how metals are absorbed by host epithelial cells in the gastrointestinal tract. And so looking at iron first, it's probably the most well-studied metal with regards to the gut microbiota, and it's probably also the one that we know the most about in terms of dietary absorption. It's tightly regulated, which we know is important because free iron can generate the production of reactive oxygen species, which are toxic. And also a lot of the dietary iron is present in the ferric form, which we know is very insoluble. And so although there's around 3 to 5 grams total iron in the body, only around 1 milligram is absorbed or excreted in a day. Most of the iron comes from recycling of red blood cells. And most of the absorption itself happens in the small intestine. So that's where a lot of the zinc absorption also happens. So I said that a lot of the dietary iron is ferric. There's also heme iron that can come from the diet as well. And so the uptake pathways that I've highlighted here are for ferric iron. There's a protein duodenyl cytochrome B that can reduce the ferric iron prior to its being taken up across the apical membrane of the epithelial cell or enterocyte by DMT1, which is a transporter that uses a proton-coupled mechanism to take up iron too, and it can also take up other divalent metals. Another protein transporter, HCP1, can take up heme. Once heme gets into the cell, it can be degraded by heme oxygenase to release the iron. In the cell, the iron can go and perform its functions with different proteins, or it can be stored in ferritin. And then iron can be exported by ferroportin, or FPN1, out of the cell across the basolateral membrane towards the bloodstream. It can be oxidized by a copper oxidase protein and bound to transferrin. And so it's tightly regulated. So there's a few mechanisms of regulation that I want to highlight here, especially because recent studies have shown some interesting things regarding how the microbiota affects this. And so under iron deficient conditions, HIF2 alpha, which is the hypoxia inducible factor 2 alpha, is stabilized, and it actually promotes transcription of the 3 proteins. So D-site B, DMT1, which facilitate uptake of the iron, and FPN1, which facilitates exporting it across the basolateral membrane. So those are increased under iron deficient conditions. So basically promoting transcription of the iron uptake genes. Low iron in the cells also results in increased RNA binding activity of iron regulatory proteins, so IRP1 and 2. These will repress the translation of ferritin, so that's the iron storage protein that I've drawn here. And so it will reduce iron sequestration in the cells when they don't have a lot of iron. There's also systemic iron regulation. So a hormone, hepcidin, will actually over here bind to and degrade the ferroportin 1. So this prevents iron export and helps reduce the import of iron into the enterocytes when there's higher iron levels. And recent work, which I'll also talk about a little bit later, is that some species of the microbiota, like lactobacillus, will make metabolites. So I've shown here ruterin and 1,3-diaminopropane that actually affect the function of HIF2 alpha. And so we'll talk about that a little bit later. All right, and depending on the forms of iron and the microbiota, there's probably also pathways and import pathways and regulatory pathways that are yet to be seen. So in terms of zinc, zinc is also tightly regulated. There's a high amount of it in the body, though a little bit less than iron. And like iron, most of the absorption happens in the small intestine. It's mainly mediated by ZIP4 and ZNT1, so two transport proteins. ZIP4 will take the zinc across the apical membrane into the cell, and ZNT1 exports the zinc where it can go and likely bind to albumin. ZIP4 is regulated by dietary zinc, and so under deficient conditions, the mRNA is stabilized, and so the protein will accumulate. And when there's higher zinc, then it gets degraded. There's also transporter proteins that will actually transport the zinc back across the basolateral membrane. So ZIP5 and ZIP14 have been identified to do this. And then ZNT5B will transport the zinc back into the intestinal tract. And so ZIP5 is actually translationally inhibited under deficient conditions by a polysomal stalling mechanism. And so when the zinc is repleted, so you have more zinc back, it can be rapidly expressed. And then ZNT1 transcription, so this one that exports it across the basolateral membrane, that's regulated by MTF1, which also regulates metallothioneins that store zinc in the cell, and it senses cytoplasmic zinc. And then just zinc in the cell can go to the intracellular organelles, the proteins it needs to go to, and metallothioneins. All right, and so that's sort of our summary of zinc uptake in epithelial cells in the gastrointestinal tract. So this is from the host perspective. I didn't really talk much about the microbiota here yet. I don't have specific studies that have shown a direct interaction, but as we've seen for iron, it's possible that they're there and we just haven't identified them yet. And then one other thing to note is that different dietary forms of zinc can affect its uptake. So one that's known is that zinc bound to phytate can actually inhibit its uptake. So that will affect things as well. So of course, bacteria look quite different from eukaryotic cells. They're a lot smaller. They don't have the intracellular organelles that you see in the eukaryotic cells, and they have different cell wall architectures. So if you remember, there's gram-positive and gram-negative bacteria. Gram-positive bacteria just have a single cytoplasmic membrane, and they're encased in a thick peptidoglycan layer. Whereas gram-negative bacteria have two membranes, the cytoplasmic membrane and the outer membrane, and then the periplasm in between. And there's other cell wall architectures, proteins, and other things that are bound to the bacteria. Some of these can affect certain species' ability to adhere to the mucus layer, as I mentioned. But I'm not going to talk too much about those in detail here. So this is just to remind you of the differences between gram-negative and gram-positive bacteria. And so to talk a bit about bacterial metal homeostasis, I think you've heard some of these things before, but regarding iron, it's generally not very available. And so bacteria that need it have high affinity transport systems. So you've seen this with siderophores that Allison talked about. So that's one way that they can scavenge iron from the environment. They can also store it in their cells and use it when they need it. They can control iron consumption by downregulating iron-containing proteins. And then, of course, there's regulatory systems that can coordinate all of this machinery in response to the availability of the iron. And so if we just focus on iron uptake, looking at gram-negative bacteria, I have this figure from a review paper. There could be passive diffusion, but given that iron is scarce, that's likely not a big player here. As I said, bacteria can chelate the iron using things like siderophores, and they can also take up iron chelated to other things like heme. And so on the outer membrane here, we just have transporters that's taking up iron in various forms. And they do this in an energy dependent process that requires the ton B complex, ton B XBB XBD. And then the complex can go in and bind to an ABC transporter and be transported across the inner membrane into the cell where it can undergo further processing to get the iron. Some bacteria, ferrous iron uptake is more important under anaerobic or microaerophilic conditions. So I've highlighted one here, a transporter called FeOB, which has been primarily studied from E. coli. And then when you look at gram-positive bacteria, they don't have that outer membrane, so they can bind directly to the ABC transporter. And there's also been other systems identified, such as the one that I'm showing here, which is an iron regulated surface determinant system, so ISD, which binds heme and can transport it down and across into the cell. Okay, so that's the summary of iron. And now for a quick summary of zinc. So of course, cells that need zinc can have high affinity zinc acquisition systems as well. There's intracellular buffering mechanisms. We saw a lot of this, David Giedrock talked about recently. We can have zinc sparing responses where we're not using the proteins that require zinc and instead using other proteins that do the same function that don't require zinc. And then there's regulation, just like with iron. So if we look just at the uptake systems in gram-negative bacteria, the high affinity, or I'm showing it in gram-negative bacteria on the left, gram-positive bacteria on the right. And so there's high affinity acquisition by an ABC transporter, so ZNU ABC, and there's ones that look a lot like this in nearly all bacteria. So the one in gram-positive bacteria shown here is ADC ABC. And so we can have zinc that binds to the substrate binding protein part of the ABC transporter here. Some have this ZinT, or it's a periplasmic zinc binding protein that can help facilitate uptake of zinc by the ABC transporter. And there's now some evidence that there are also outer membrane transporters that resemble the ton B ones that I just showed for iron. And then, so this is high affinity zinc uptake. There's also low affinity ZIP transporters. So ZUPT in E. coli is constitutively expressed, and so that can also contribute to zinc uptake. And so this is really sort of the background that I wanted to cover on metal uptake in host cells as well as in bacterial cells, to give you an idea of how they might transfer between the two types of cells in the microbiota. So I think before I move on to how metal availability affects the microbiota, maybe this is a good spot if there are any questions.
Okay, so if you have questions, once again, please put those onto the chat, and I will either call out a person or read them. Probably, since we're hesitating here for a moment, I'll just call on the person who might have it. Let me just make one comment while you're thinking of your questions, and that is that this HIF2 alpha system that Melissa described, what she described in terms of metal ion uptake in the gut, but that was the subject of the 2019 Nobel Prize in Medicine, because this is the mechanism by which oxygen sensing occurs in humans and is critically important in things from ischemia to cancer. So Catherine Siller, sorry, Siller, has a question.
Hi, you're talking about how there was some evidence of proteins similar to ZNT for the outer membrane. Would those be like attached to the membrane, or would they be like out or
Oh,
not?
so like ZNT, so actually I think what I meant there is that it's proteins that are like the, so on the outer membrane, I've shown ZNUD here. This is analogous maybe to the ones that I showed for iron on the previous slide. So let's see if I can, there are 2 slides ago. So here, these systems, which take up the iron chelates in an energy dependent process. So this is the actual transporter that's embedded in the membrane that it can go through. And then this is the TON-B complex, which probably goes somewhat across the periplasm and then has components bound to it in the inner membrane. And so that gives the energy for transporting the iron complex and potentially also zinc now across that outer membrane so that it can get in and get to the ABC transporter to be transported across the inner membrane.
Gotcha.
Does
Okay.
that answer?
It does, yeah.
Great.
So Melissa, I'm going to just follow up on something close to that. You were saying that zinc has a ton B like transporter for it. And normally when I think of ton B, I think of ferric enterobactin. Is the ton B like zinc transporter something which transports hexaqua zinc, or is it taking a chelated form of zinc and transporting it through the membrane?
That's a good question. I don't know too much about the details of how it works, except that there's some evidence. I don't think there's much. There's really a lot less known, it seems, about zinc than iron, but that there's some evidence for an analogous process for the zinc. But there's really, I couldn't dig up a whole lot of information on it.
Yeah, the iron has been the king for so long,
Yeah.
but now people are recognizing the importance of zinc, which, given what you were saying, that iron is one of those things that once the body gets it, it really doesn't want to give it up. But I thought it was interesting when you were talking about this sort of flow forward and flow backwards of zinc through the basolateral membrane and back out into the gut. You'd never see that with iron.
Right. Yeah. Yeah, exactly.
Shreya, you have a question.
Hi. So you showed that zinc in its phytate form prevents its absorption. So I just wanted to ask if it's the same for iron, or are there other forms of iron in the diet that prevent its absorption?
So there's probably, so I guess iron absorption, I didn't really say it in too much detail, but it's actually very low. So a lot of the iron that comes from more plant-based diets is the ferric iron, and percentage uptake of that probably ranges anywhere from 5 to 15%. It's very, very, very low. And so not a lot actually gets taken up. I don't know specifically about phytate. Most of that I've seen in terms of zinc, but there's lots of dietary components that the metals are binding to, because they're not going to be free hexaqua in the gut. There's too much other stuff around, at least not out floating. They may get converted that way and when they go through their transporters, but yeah, a lot of the iron in the diet is not taken up. It actually goes right through. Yeah.
Thank you.
Olivia?
Hi, Melissa. I'm going through your thesis and everything, so it's so nice to meet you finally. So, my question was that you were talking about the gram-positive bacteria has like siderophore binding proteins to bind the iron siderophores. So like, how is an iron siderophore imported when an apple siderophore is initially bound to the siderophore binding protein? I was
So
reading.
how is the iron bound to the siderophore actually imported? Is that the mechanism? So
Yeah, like because when the aposiderophore is already like binding to the siderophore binding protein, how is the iron siderophore being included?
So you mean across like the inner membrane here? I guess I don't know a whole lot of details about that other than that it happens.
Yeah, so Olivia, I think it depends quite a lot on what type of siderophore you're talking about. Some siderophores donate their iron to siderophores that are associated with the membrane, like in the mycobacteria, but others, like Allison talked about, and she was online, she might want to clarify things after I say this, but the iron is taken, is bound by the siderophore, ends up going to a outer membrane transport protein. I'll talk about the gram negatives right now. And then it gets pushed into the periplasmic space where that siderophore can be taken by a periplasmic binding protein to one of these ABC binding cassettes. It goes through the membrane, and once it's into the membrane, depending on, once it's into the cytosol, excuse me, then the iron is removed. Now, if it's a hydroxamate siderophore, those usually have reduction potentials where you can have direct reduction. If it's something like a catechylate siderophore, what you have to do is have an esterase that cleaves it. Professor Butler talked about that in the previous lecture. And when that cleavage of that that serine, or if it were bacillibactin threonine backbone occurs, you see about a 400 millivolt shift in the reduction potential. And that allows the iron 3 to iron 2 reduction to occur in the cell. Plus, it means that the catechylate siderophore, Enterobacter, for example, when it gets all destroyed, isn't going to be pulling iron out of places it shouldn't be pulling it out of in the cell. Remember, these are the biggest apes on the block, so they bind iron whenever they want to bind iron. So it's a complicated thing. I would recommend you go back and look at some of what Professor Butler said in the first lecture. Those are up on the Canvas site.
Thank you.
Okay, it looks like we've gone through all these questions, so why don't we move on, Melissa?
Sounds good. All right, so with all this in mind, we can now think about some of the different factors that can affect where metals are and how much is there in the gastrointestinal tract and in the microbiota. And so there's really a complex interplay here between us, the microbes, which there's also an interplay there between commensals and pathogens, as well as the things that we take in. So the diet and antibiotics and drugs. And so metal availability in the gastrointestinal tract is affected by how much is there from the diet. Antibiotics can affect it, genetics can affect it, and host-imposed metal withholding or overload can also affect how much is there. So this is that nutritional immunity thing that I know you've already heard about, and I'm sure you'll hear about more from Peggy. And so the results of changing metal availability in these various ways is that it can affect the commensal versus pathogen colonization. So if you throw off the balance of the microbiota by giving it different amounts of metals through antibiotic treatment or diet, it can throw off the balance of things, so it may not be as good at protecting against pathogens. It can also affect the metal processing. So what metal homeostasis mechanisms are used by the host or by the microbes in the microbiota. So given what different forms of metals are present, there may be different uptake and transport pathways. And then metals that are there can affect activity of metalloenzymes that are there. So depending on which metals the metalloenzymes need, that can affect it. And then microbial metabolite production can also be affected. And so this can happen in various ways if the enzymes that make the metabolites are affected, or if the metabolites themselves can bind metals, or if their activity is affected by metals. You can see results from that. And so thinking a little bit more about the complexity of this, I've now shown both the iron and the zinc uptake pathways in the gastrointestinal tract that I already talked about. And what I want to draw your attention to here is the mucosal side. So it's meant to represent the mucus in blue, and then the lumen of the intestine in this pinkish color. And so there's really a complex interplay between our cells and the microbes that inhabit these mucosal surfaces. And so their access to metals really depends on the mucosal environment. So what metals are present and what their bioavailability is, this can be affected by the dietary components. So, zinc bound to phytate, as we mentioned, or iron bound to different siderophores. And so that may affect what acquisition pathways are used by different bacteria. There's also potentially some mucosal secretions in the form of metals from those is not really known. And in general, the prevalent form of metals on the mucosal surface is not well defined. So I think David mentioned last week about metal speciation. And so that's something here in the gastrointestinal tract that you can imagine can be quite complicated. And it's just not really been looked at in a lot of detail. And so that complicates things. And so in terms of metal microbiota studies, a lot of them have really focused on just identifying what's there. So a number of animal studies, in the case of iron, some clinical studies, and also in vitro gut model studies tend to look at what happens if you give excess metal or put it under deficient conditions. What's there? How does the microbial composition change? So what groups of bacteria either go up or go down, and what species go up or go down? So relative changes are what is usually measured here using sequencing methods. And so you basically just see what changes under metal supplemented conditions or metal deficient conditions. And so I'm going to give a summary of sort of where these studies are at for both iron and zinc. In the case of iron, which has definitely received more attention, I'm going to preface this with saying that the outcomes of these studies are complex, both for in vivo and in vitro. And so that's because the host status matters, and also because the form of the iron matters. So in terms of the iron supplement. But there are some relatively consistent findings that have been learned from these studies. And one is that when iron supplements are used, there's generally an iron-induced decrease in beneficial groups of bacteria. So this is like lactobacillus and bifidobacteria. So those you may have heard of, because if you look on the ingredients of probiotics, they're often there. And they're present in your gut as well. And so it's consistent to see a decrease in those groups of bacteria towards more a group of bacteria and enterobacteria that contain more of the opportunistic pathogens, such as E. coli and Salmonella. So that's a relatively consistent finding. However, the host status does matter. So there are some studies that have seen, the former case where there's a decrease in lactobacillus and bifidobacteria, but they didn't see an increase in enterobacteria. And one of the hypotheses on that is that the subjects of the studies weren't in very diseased areas. And so they didn't have a high enteropathogen burden to begin with. But this is quite complex. And so there's a lot of factors that can affect it. And even though there's a number of studies on this area compared to zinc and other metals, compared to other nutrients, there really aren't a lot of studies. And so there's a lot more to be learned here. And so that's sort of the summary from the iron supplement side. There are some animal studies that have measured what happens with zinc deficient or iron deficient diets. And one thing that's been found a few times is that there's an increase in lactobacillus. And so this group of bacteria is actually interesting from a bioinorganic perspective because it's often cited for having low iron requirements. Same with the Lyme disease pathogen. And so usually chelating iron from some of these species doesn't affect its growth. There's a lot of different species of lactobacillus, and there's some factors that make this interesting and worth looking at a bit more. But this would be consistent with low iron requirements of lactobacillus that they could maybe increase under deficient diets because they don't need as much. Okay, and so that's sort of the summary of iron. If we look at zinc, there's fewer studies, but we've also seen some, there's a few things that we've learned. And so one of the things that's known is that zinc is important for the integrity of the mucosal barrier. And so I have a figure from a review paper here, just showing the epithelial cells. I've got the goblet cells, which again, those are the ones that make the mucus, or the mucin that makes the mucus layer. I've also highlighted here Paneth cells. And so these are cells that are important because they make antimicrobial molecules that can help sort of keep the bacteria from invading. Interestingly, they also have zinc in their granules, and they're susceptible to zinc deficiency. And so zinc is really needed for the function of those cells. And under zinc deficient conditions, we basically get barrier dysfunction where the bacteria can cross, and we have bad gastrointestinal conditions such as diarrhea and inflammation and things. And zinc supplementation helps with these. In terms of the microbiota, deficiency has in animal studies been correlated with decreasing levels of Firmicutes. So this is the group that includes a lot of the beneficial bacteria. It's a phyla. It's one of the major phyla in the gut microbiota. And so deficiency correlates with decreased amounts of bacteria within that phyla and increased amounts of bacteria within proteobacteria. So that's the one that it includes a lot of the pathogens. And so deficiency just shifts the microbiota to one that has more pathogenic potential. There's an interesting study where they supplemented a broiler chicken model that was infected with S. typhi. They supplemented it with zinc and found that zinc supplementation increased the beneficial bacteria while decreasing the detrimental ones. And so zinc supplementation seems good, but you also don't want to go too far. There is an interesting study that came out of the SCAR group at Vanderbilt that showed that excess zinc in mice actually reduces the diversity of the gut microbiota, and it rendered it more susceptible to Clostridium difficile infection. So this can often be induced by antibiotic treatment, and less antibiotics were needed in the zinc excess mice. Okay, but in general, there have been these few animal studies, but there's really not much clinical data like we saw with iron on how the gut microbiota is modulated by dietary zinc. So there's a lot to be learned in this area. And so, and in general, a lot of this summarizes, we're just looking at what's there. So there's all these different species within the groups of bacteria. Within the species, there's a lot of different strains. And so this is just asking, if we change the metals, what's there as a result, but it doesn't tell us anything about the underlying mechanisms and why those relative levels are changing and what the bacteria are doing. And so what are some mechanisms that might underlie these changes? Of course, one is that there could be direct effects on the bacteria. So different bacteria may have different nutrient requirements. For example, lactobacillus, as I mentioned, which have relatively low iron requirements, they have pretty high manganese requirements. They will take up a lot more manganese than other bacteria do. But of the hundreds of species that are present in the microbiota, most of them have not been studied from the bioinorganic perspective, and we have no idea exactly what their requirements are. as well as their homeostasis mechanism. So what sorts of transport proteins they have. There's a lot of genomic data now, but a lot of it just hasn't been analyzed, and the metal transporters have not been looked at in much detail. So there's probably a lot to learn there. There's also indirect effects that may impact the metal bioavailability and then potentially also affect what's there. So I'm going to give a specific example that's related to one of the papers that I assigned on how different metabolic processes might affect how much metal is there, and then that can affect different species' ability to survive in the microbiota. Of course, metabolites are important. So as I said, they can maybe communicate with the homeostasis mechanisms, or it's possible that some metabolites can bind directly to metals and affect their functions. There's also bacterial signaling. So bacteria make small molecules that allow them to sort of communicate with each other. And there's maybe some evidence that metals can have effects on that mechanism. And there's certainly some metal proteins that are involved in synthesizing some of those signaling molecules. And then colonization is another thing that might be affected by metals. And so I talked about the mucus layer earlier, which again is this glycosylated polymeric protein. And it turns out it does have an affinity for metals. And so there's some older work and some recent work. It's a relatively low affinity, like in the micromolar range for zinc, but there is some capacity there. And the bacteria, the ones that can adhere to the mucus layer, have surface proteins that can bind to it. And so it's possible that metals can be affecting this interaction potentially, either facilitating if they can bind to some of these proteins, that's possible, or maybe inhibiting it. And so changing the amount of metal that's present in the gastrointestinal tract could also indirectly affect whether or not the bacteria can colonize. And so I'm going to go through some specific examples where some of these mechanisms are maybe starting to come to light. So this one's related. Actually, maybe that's a good spot to stop for questions if we want.
Okay, so you know how we work this with the chat. If you have a question, please type it into the chat. And William shot right in. He has a question, so go for it, William.
So I know, so you talked about how metals might have help stabilize the mucous membrane, basically, right? I was just curious. I know that mammalians secrete MMPs. I was wondering if bacteria had anything similar that might also affect the mucosal membrane.
That might affect the structure of the mucous membrane.
Yeah.
So some bacteria will actually degrade it and use it as nutrients, essentially. So yeah, there's enzymes that they can make to break it down. Yeah.
Thank you.
Garrett?
Okay. Hi. So my question was more about the Panth cells, and what sorts of antimicrobial molecules do they produce, and then how are they affected?
Peptides, I believe. Yeah, it's like small peptides. But I don't know too much of the details. So it's, I've focused a lot more on the bacterial side of my own research. But I think it's mostly small peptides, but there's, I think, a lot to be studied in that area, especially with the zinc. Like, we know that the zinc's there and a little bit about the transporters, but not really a lot about what it's doing either.
Okay, thank you.
Yeah.
So Melissa, I have to admit, I was really intrigued by this discussion that you had regarding how zinc ion levels affected basically the integrity of the membrane, and so that bacteria couldn't get through. I think so much, or I think many times we think of the bacterial colonies that one has in the gut associated with things like gut diseases. So diarrhea,
Yeah.
for example, Clostridium difficile, salmonella infections. But there has been a lot of talk recently that diseases like Alzheimer's may actually represent cases where anaerobes in particular have been able to escape the gut, get into the bloodstream, and then cross the blood-brain barrier, and that the damage in the brain is coming from microglia that serve as basically an immune system in the brain to try to go after these organisms. So has anybody followed up on that end of the microbiota in other disease states in humans, and whether that could be correlated with zinc um, levels?
Whether it could be correlated with zinc levels, I would say I have not seen anything specific to that. There's really a lot of room for discovering a lot of things here. Regarding that in other diseases, I mean, I do know that if the barrier is compromised, this can be the cause of a lot of like inflammation and just gastrointestinal diseases, because the bacteria can sort of get through and you have a response to that. But I don't know in terms of any other specifics, and especially not with metals. And there's been very few studies on zinc in the Paneth cells in general. There's been zinc, ZP1 was used to see that there's zinc there, but really not a lot else.
Very interesting.
Yeah.
Okay, well, let's see. It is 3:25, so we've got about 20 minutes.
Okay. All right. And so I'm going to go through a little bit now on some specific examples that highlight some of the underlying mechanisms. So this was from another paper that I had suggested reading. And so I think that this example shows how the microbiota can adapt to metal availability, or what the host metal status is. And so this research group isolated some bifidobacterium strains from the gut of iron deficient infants. And they used an iron deficient medium to do this. They isolated a bunch of the strains. And then they tested them for both siderophore activity and their ability to take up iron, and compared it to well-studied type strains, or strains that came from culture collections. And what they found is, so I have siderophore activity on the left and iron uptake on the right. And they found really strain-dependent results. And so siderophore activity was detected in essentially all of them, but some could have much higher siderophore activity than others. And then the same thing for iron uptake. There was no correlation between the siderophore activity and the iron uptake, but in general, the strains with the highest siderophore activity did come from the new isolates that came from the iron deficient infants. And some of these strains weren't affected if they were treated with iron chelators, so their growth wasn't affected by that. So what they did was they took two strains. They took the one that had the highest iron internalization and the one that had the highest siderophore activity. And in an earlier study, they checked how well these strains could inhibit the adhesion of enteropathogens to an intestinal cell model that could secrete mucus, and found that the one that had the highest siderophore activity, now to be clear, these both had siderophore activity, but the one with the highest could reduce the adhesion of both S. typhi and pathogenic E. coli, while the one with the highest iron internalization was only able to inhibit S. typhi. And so in a later study, they analyzed the genomes of these strains and compared them to type strains or culture collection strains. And essentially the genome contents showed that there was some evolutionary adaptation to the low iron conditions that they had to survive in. And so using this comparative genomics, they identified some possible siderophore biosynthesis genes with an ABC transporter, as well as both ferric and ferrous iron operons in the strain that had high iron internalization or uptake. And then in the other strain with the highest siderophore activity, they found that there was a ferric iron operon that had likely been acquired and wasn't in its original genome, as well as a ferrous iron transporter. And then experimentally, what they did was they grew these strains in some iron deficient medium, and they collected the supernatant to see what proteins were there. And so this is secretome analysis. And what they found was that for the high siderophore activity strain, they identified a ferric iron transporter, which matched the genome analysis, but they didn't detect any ferrous transporter. These are, I should point out that bifidobacteria are obligate anaerobes, so they have to be grown under anaerobic conditions. And then for PV20, they saw something different. They saw, this is the one with the high iron internalization, 3 ferrous iron transporters, and they found an ABC transporter that had been linked to the putative siderophore genes. So 2 different uptake mechanisms under the same growth conditions were being used by these 2 strains. In both, they also found a zinc transporter. So they found the binding domain of ZNU ABC in both of the strains grown under the iron deficient conditions. And they also found ZIN-T. So that was the periplasmic binding protein that I mentioned that could facilitate zinc uptake by a ZNU ABC transporter. And they found that in one of the strains, so the one with the high siderophore activity. And so I think this highlights something that we really have to consider, which is even when you look at a single species of bacteria in the gut microbiota, strain to strain, there could be very different metal activity by them. And so they could have very different competitive functions in being able to survive under changing metal availability. And so this complicates things and also offers an interesting thing to pursue and really shows that there's probably a lot of uptake and transport pathways that we have to define and discover. Okay, and so the microbiota can also modulate metal homeostasis. I'm going to give some examples here for iron, one of which I already mentioned. There's a report that shows that the microbiota can also alter host iron sensing. So using germ-free mice. So these are mice grown in conditions where they won't develop a microbiota, and then you could colonize them with the bacteria that you want to study. They found that the iron uptake proteins, so D-cyp-B and DMT1, are less abundant in those germ-free mice. But when they colonize the mice, the cells then favor iron storage. And so this is potentially a way that the microbiota could inhibit growth of pathogens. So if they favor the enterocytes to store that iron, then it might be less available to the pathogens that are present in the gastrointestinal tract. There's also some very recent evidence that the microbiota can affect the systemic iron homeostasis. So this is the homeostasis that's regulated by hepcidin. So there's some links there that the microbiota can determine the extent of the hepcidin induction. And then intriguingly, a group from Michigan reported what I mentioned earlier, which is that the microbiota can, some members can make metabolites that can modulate local iron homeostasis. So they can produce these things like ruterin and 1,3-diaminopropane, which reduce expression of the iron uptake proteins, and they induce iron storage proteins. And so under iron deficient conditions. And so these iron deficient conditions may select for a group of bacteria that can do this. And then there's fewer examples with zinc where we've started to define mechanisms, but I thought that this was an interesting one to cover. because it shows how the microbiota can modulate metal availability. This also came out of a group at Michigan several years ago, and they were studying a commensal organism in chickens. It's pathogenic to us. And they found that it required high affinity zinc transporter, so ZN-UABC-like, in order to survive in the presence of a normal microbiota. But when the chickens were germ-free, this high affinity zinc uptake transporter wasn't needed. And so they went searching for the mechanism. They measured the amount of zinc in the intestinal contents of the germ-free chickens and found that there was less overall zinc there. There's also less iron and magnesium and more copper and manganese, but there's no discussion or anything on the other metals at this point. But they took those intestinal contents and prepared medium based on it. And there was still growth inhibition if you took, in the absence of the zinc transporter, when it's grown with medium prepared from the normal microbiota chickens. So they took that and ran it on immobilized metal affinity chromatography, and they were able to isolate some zinc binding proteins. Those, when put in media, would inhibit the growth as well, unless the high affinity transporter was there. And mass spec of those proteins showed that most of them were digestive proteins that were known to bind zinc, so things like aminopeptidases. And in fact, in the germ-free chickens, many of those proteins weren't present. And so there were different metabolic capabilities in the presence of the microbiota and in the absence of the microbiota. And so through those, there were different proteins present, so different enzymes that have different metal requirements. And so this led to different metal availability in the microbiota ultimately. And so that's an example of an indirect way of affecting the bacteria and which ones can compete well enough to survive. All right, and so before I talk a little bit about the tools and whatever time I have left, I just wanted to put up a little summary of sort of what I've been discussing, which is that we know metal uptake influences microbiota composition, as well as susceptibility to pathogen colonization. And we've seen that the microbiota can modulate metal availability and host metal homeostasis. So not only does metals affect the microbiota affects the metals. There's a lot of open questions in this area, which I hope has become apparent. And I've highlighted some of those here, like what are the metal requirements of different gut microbes? Are there different homeostasis mechanisms being used by some of these commensal organisms? How do those impact microbe-host interactions? What is the speciation of the metals in the intestinal tract, and how does that change? And then overall, how do metals influence the microbiota function? So different species may have the same function, and so it's worth looking at on a more global perspective as well. And so in the final section, I just want to touch on some methods that we can use to study metal trafficking and metal homeostasis in the microbiota. So of course, you saw that there are so many genes, many of these are not characterized. And so of course, doing transcriptomics and proteomics studies under different conditions is an area that is likely to produce a lot of interesting results. Similarly, doing metabolomics and trying to identify some of these metabolites and how they interact with metals is important. But we can also think about doing direct studies where you directly detect the metal and try to see where it's going, how much is there, and what it's doing. And so we can do this using optical metal sensors like fluorescent metal sensors. And this is the area in which my group is focused on. And so I just want to touch on this. So you can have different families of fluorescent sensors. We can have synthetic sensors, so molecular probes, and I'm just showing some examples here for zinc, or we can have genetically encoded fluorescent sensors. So these are based on proteins, in particular fluorescent proteins. And so in terms of studying the gut microbiota, what we really want to be able to do is to study the community of bacteria. So multiple different species because it's likely that a single species on its own is not going to behave the same way as it will in the presence of other competing organisms or even in the presence of the host. And so molecular probes are really, really useful, but they won't allow us to target to a single species of bacteria within a community. And so we'd really like to be able to use genetically encoded probes that we can put, have the bacteria make that sensor itself. Of course, that means we need to be able to genetically manipulate the bacteria that we're looking at, but we can do that for some of the gut microbes, not nearly all of them, but some. But the other problem is that a lot of the existing genetically encoded sensors, which we have for calcium, zinc, and copper, are based on the green fluorescent protein. And the green fluorescent protein and its family members require oxygen to become fluorescent. And I haven't really specifically discussed this, but there's very little oxygen in the gut microbiota. And most of the organisms present there cannot grow in the presence of oxygen. And so this means to study live cells, we need fluorescent proteins that are going to work in the absence of oxygen. And so there's been increasing attention on these cofactor binding fluorescent proteins for various reasons. And there's a number of different examples. And so these are just proteins that bind external cofactors. So they don't require oxygen when they fold to make the fluorophore within the protein, but they can bind these cofactors even in the absence of oxygen and become fluorescent. And so there are flavin binding fluorescent proteins, which bind flavin mononucleotide, and they're primarily green fluorescent. They're also nice because they're about half the size of GFPs. And then there are a number of bilin binding proteins. So these bind linear tetrapearls. They're essentially breakdown products from heme. And so the near infrared ones have probably gotten the most attention because they're in the near infrared, and none of the GFPs go quite as far over in the spectrum. But there's also cyanobacteriochromes, which can pretty much cover from blue all the way into the near infrared. And so there's a whole wealth of proteins here. Those are all natural cofactors that can be made within the biological system. There's also examples with synthetic cofactors that can bind to engineered proteins and be fluorescent as well. And there have been a number of examples where these are starting to appear for calcium sensors, though there are none for zinc yet. And so we've been exploring aspects of many of these proteins in my group, and hopefully we'll have more published this year. But I just want to highlight one of the first ones that we worked on, which is published, which is one of these flavin binding fluorescent proteins called Kralove, which of that family is the brightest one with the highest extinction coefficient. And one of the first things that we wanted to do was just see if mixing this protein with metals affected its fluorescence, because building a metal sensing platform on it requires understanding how the fluorescence responds to metals, before you put in a metal binding site. And while most metals don't affect its fluorescence, at least in the absence of a histidine tag, copper does quench the fluorescence of this protein, and it seems to have a low micromolar affinity. And we think we know where a weak binding site might be in it, and we've done some studies since then, but we can essentially use this as a weak copper sensor under live cell conditions as well. And so here I've just shown the fluorescence intensity when you express this protein in bacteria. We're just doing E. coli here. We add a chelator, an intracellular chelator, to chelate any copper that might be in the cells. This is copper 2, so there's probably not much. And then we add a bunch of copper 2 into the cell, and we see the quenching. I also have lifetime over here on the right. So that's another way of detecting fluorescence that could be very useful for single fluorescent protein sensors, because it doesn't depend on the amount of protein there and may allow us to quantify the amount of metal. And the nice thing about the flavin binding fluorescent proteins is they have relatively long lifetimes compared to GFPs. And so they're potentially very powerful in this area, and we do see a change in the lifetime when we add the copper there. And so we're working on building platforms for sensing metals such as zinc, and hopefully I'll have more to share about that in the future. But with that, I'm going to end here, thanking my group. So I have an old picture and I've appended all the new members around the side. And so they're the ones really doing most of the work now. And then of course, I want to thank all of our funding from the Welch Foundation, the NIH, and UH, of course. And thank you all, and I'll be happy to take any other questions.
Well, thank you very much, Melissa. Clap, 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 clap. I received a direct message from Allison Butler, who had to leave just a couple of minutes ago. And she said, wow, this is fantastic. Lots of things to do here. And I think that's absolutely, absolutely true.
Yeah.
I'm going to ask one question before we go to the student questions. It's a tough question to really kind of answer, but I think it's an important one just so that people keep a perspective on this. And that is that, you know, when you talk about the gut microbiota and such, and I realize you're looking at chickens. Okay, you're not looking at square chickens, but you're looking at chickens.
Yes.
But ultimately, we want to translate this back to humans.
Right.
And what I would say is that it's not just even humans. Has anyone even attempted, maybe through feces samples or some other way, to see if there are differences in the microbiota with race, with changing age, with gender, all of these issues? I can imagine hormonal levels
Yes.
impacting things. I mean, not even to mention what happens if someone gets sick, but just these others. Maybe you could tell us a little bit about that before we go to some of the other uh, questions that are here.
Yeah, that's a really excellent point. And there are, you know, there's certainly studies. I have not organized them in my literature searching, but definitely age, all those factors can affect what's present there. And, you know, even just where you live in the world affects what's present there based on diet and other environmental factors. And so, and there's different species. You know, I said there's hundreds of species, but when you go from one person to another, they're not necessarily all going to have the same species. There's ones that show up in a lot of people, but it's not necessarily the same. And so that really does complicate things. But I think, and I only very vaguely touched on this, I think that highlights why it's important to understand the functions of the microbiota as a whole as well. So just because one species isn't there performing whatever function is in its genome doesn't mean that another one hasn't replaced it to do the same thing. And so you'd really need to do studies to analyze what they're all doing together to see what functions are covered. But yeah, it's different from, it really is complicated when you start digging down.
Yeah, no, I mean, but of course, because of that complication, it could be extremely, extremely important.
Yeah.
So what we're going to do is go to some of the questions. Shuri, you had one?
Hi, so since the microbiota is dynamic with not just one static climax community, in metal deficient conditions, is there evidence that bacterial metal acquisition genes are regulated by transposon activity?
I do not know. I have not seen anything regarding transposon activity where that's been looked at, but that's a good question. Yeah, I really just touched on the studies where they've even just started to look at what transporters are there in what species. But we're really just scratching the surface at this point. So
You mentioned that some of the genomics are being done on these, but how many? I mean, okay, lactobacillus has been around for a long time. I mean, all these bugs have been around for a long time, but we have recognized that they've been around for a long time. Do many of how many of these organisms actually have a complete genome sequence that one could go through and look for transposons, for example?
there's a number of, and the genome sequences are, increasing. There's really a number of them that are out there. It's just a matter of going through them and looking for these things. And I think the bioinorganic community has not paid much attention to this. Even lactobacillus, right, which have been, and this is one area that my group's working in too, is looking at lactobacillus. We've cited them in the literature so much, but a lot of that work was done before we knew the genomes, and people haven't looked very closely at their genomes. And lactobacillus is actually an incredibly diverse group of bacteria when you really look at them. And so there's just, even with the bacteria that we know some stuff about, there's a lot that we don't know once you start digging, now that we have genomes reported for them. And for the genomes that are reported, you know, there's a lot of annotations, so you can dig through those, but they haven't been studied. It's just, you know, uncharacterized protein or putative ABC transporter and things like that. And so there's a lot to explore.
Catherine Hunter.
Hey, I was kind of wondering with the last portion about the cofactor binding fluorescent proteins.
Sure.
Would there be an issue with maybe using them in live cells because the distribution of metal ions is so tightly regulated, especially in human cells?
Well, so the idea, I guess I didn't talk about what metal we're specifically hoping to use them for, which is zinc. And the idea of using them would be both in cells, but also appending them to the surface to look at the metal environment around the cells. And so in bacterial cells, and David actually mentioned this last week, that nobody's really measured free metal levels in bacteria inside their cells directly with metal sensors. And so that does need to be done and be able to see how that might vary, because we've done it in mammalian cells, but not in bacteria. And so it would be a way, you know, you can modulate the metal affinity. We know how to do that by changing ligands on the metal binding sites to see different pools of metals that are present there. But yeah, that's a definite consideration. And it's always a consideration with sensors, that when you're putting the sensor in, are you interrupting the metal homeostasis? But you can do a lot of controls to sort of see what might be real and what isn't. Did that answer your question?
Oh, sorry.
Yeah.
Okay.
William.
Follow-up question?
Oh, no, okay, Catherine, go ahead. Then we'll go to William.
Actually, it was me. Sorry. Peg.
Oh, it's Peggy. Sorry. who are you?
I try not to interrupt the student questions, but I just wanted to follow up on the question. When you're looking at these binding experiments, are you eventually planning to look at them not just with single organisms, but with combinations of them? Because, you know, it'd be very interesting for us to discover new sort of siderophore-like things where certain pathogens, when they're living in a community, use the metal ions that other pathogens either produce or are scavenging.
Yes, and absolutely. So we're already this. Well, so this is the whole reason that we're not just using synthetic sensors. And so we're trying to use only these protein ones so that we can genetically encode them into one species within a community. And so we've started with E. coli. We're already working on some other gut bacteria, and hopefully, you know, we're going to start with just dual cultures and work our way up. And there's, you know, we're right near Baylor College of Medicine, which has a great microbiome center that has in vitro gut models. And so that will allow us to really go up to multi-species communities that look a lot like the gut, and then maybe eventually to in vivo conditions. But yes, that's the definite plan, because they're going to behave different when they're in the presence of other species.
Right. And I think that idea you were mentioning of quantitation is important too, because just for example, with E. coli, you could have huge numbers of E. coli, which you can detect with the microbiome methods we have nowadays. But some of those may have been altered, and that's more difficult to detect. Have they altered what metal ions they're taking up in the presence of a deficiency, for example? Even though the numbers are the same, their function, their homeostatic mechanisms may have differed.
Yeah, exactly.
So it's a great combination to have at once. Cool.
Thanks.
Okay, great. William?
Yeah, I was just curious if it would be possible to engineer the protein sensors to adjust their affinity for different metals, like to either improve it or make it even less sensitive.
So specificity for different metals?
Yeah.
Yeah, so most of the existing protein sensors we know from the GFPs, and there's calcium ones that use the calcium binding domain from calmodulin, and then the zinc ones actually use zinc finger binding domains. And those have been able to be modulated to bind copper as well. Other metals we don't really have much of yet. So we'd like to have iron and manganese sensors, but sort of the weaker binding affinity problems of those have made it so that we haven't made them yet. But yeah, we can certainly tune it for some metals so far, and we can change the affinity certainly for those. And that's been done for the GFP sensors. But expanding to other metals, that's an area that is still open as well.
Okay.
Clotilde Policar thanked you for an inspiring and great lecture. And the last question that I see actually listed in the chat is from Olivia.
I was just curious, like, has heavy metals any known effect on gut microbiota? Like, we know about them as neurotoxic and being present whenever there is a cancer of some sort, but like, has any effect on gut microbiota?
I can't hear everything that
I
you're
think
saying. I'm sorry.
so. Let me repeat it, partially because I've read part of it. What she's curious about is whether heavy metals, so for example, arsenic, lead, mercury, etc., whether these have any effect on the gut microbiota. And that we recognize that, for example, lead can cause anemia. It can cause all sorts of other problems when at too high a concentration. Has anyone examined the possibility that some of these heavy metals could lead to problems when people are exposed to them in the gut?
Yes, I think there have been. I haven't done a thorough literature review on them. But I know there's some stuff on cadmium. That's the one that's standing out in my mind right now. I don't know how thorough the work has been, but I do believe that there are some effects, and we might expect there to be some effects as well. But yeah.
Yeah, whenever you look at these heavy metals, of course, you know from some of the stuff we've done, and obviously David and others, it really depends on the absorption mechanism. Things like cadmium tend to be in fly ash that ends up in your lungs, and its transport, utilization, what it does is going to be very different than maybe a child that ate leaded paint that then has to absorb the lead through the gut. So again, a very complicated situation. So because I said the student questions were finished, we've gotten some questions from other audience members. And so Carl Kupke has a question for you. Carl.
Sure.
Hey, Melissa.
Hey,
great
Carl.
talk, by the way.
Thank you.
So I was curious, my understanding for some flavin proteins is that the flavin binding is actually metal dependent. We're working with one currently where like a manganese is in the coordination sphere of the flavin. And so would it be possible to engineer that metal binding site and then have it be an on switch instead of an off switch? I guess I'm curious.
Like an on switch for the flavin binding.
Yeah, I mean, I guess it depends on whether or not the apoprotein can just float around without flavin, which I don't really know very well.
Yeah, that's a very good question. We do some studies to, in order to find out how much flavin is there in the purified protein, you unfold it and measure its absorption. So I don't know that the kinetics or, of how the flavin's coming in and out in the cell are looked at. I do know that for a lot of these, you can just express them under normal conditions and they come out pretty much fully loaded with the flavin. But I think that's an excellent point on potentially trying to change, you could change the site a little bit to make it so that metal binding would result in a fluorescence turn on in that way. That would be one option of doing that. Nobody's really done that yet. So this work that we reported on Kralove, and then there's a related one called I Love that there was some work on that also showed some quenching from copper. And I think there's some quenching from some heavy metals too with another related one. But nobody's really tried to do any sort of metal turn on with this. And you could think about maybe doing it the same way that's been done with the GFPs, which is to do like circular permutation to change the fluorescence properties and insert a metal binding site that can then sort of restore the original properties. But that'll take some engineering as well. So.
Okay, yeah, if you're curious, I think there's some of these that have manganese that's required. So it might be a way to get to the manganese that you were talking about before.
Yeah.
My guess
Definitely.
is the
Thank you.
manganese quenches the fluorescence.
The manganese is there in order for the flavin to bind in the first place.
Is on the protein, but they're spatially separated so that the manganese does not. What I'm wondering, if the manganese is too close to the flavin, in theory, it could quench the fluorescence. In fact, that's what Melissa, I think, is looking at when she has these turn-off sensors.
With the copper, yeah.
Right,
yeah.
but what
true.
she
So
does.
maybe if a metal's in there just to bind the flavin, it would already be pre-quenched.
Well, if the flavin is bound to the metal, but if the metal changes, as Melissa was saying, the protein conformation, say it was like a zinc finger that could then bind a flavin, then when the metal was there, it would fold, the flavin would bind, it would turn on, and the metal would be far enough from the flavin.
Right.
That's true. Yeah, I don't know the fluorescent properties of these proteins, so it might be an issue.
It'd be interesting to see.
Yeah.
Because if it really is a conformational change and not a direct interaction, it could be exactly what Melissa is looking for.
Right. Yeah.
Okay. Well, thank you so much, Melissa. I learned a lot today. This is, I think, really an area for the future, not only in metallo-bio-inorganic chemistry, but or metallo-biochemistry, I should say. Let's not be redundant. And also obviously just for human health. I'm going to be stopping the recording here. Once again, people can get this on the SBIC site very soon afterwards. Let's see if I can actually find out where the stop the recording button went.